Hello everyone, it's Dr. Wolfe here, and in this video I'm going to be going over the process of transcription, which is going to be happening in our nucleus. We're going to start off with some DNA and some free nucleotides, not just DNA free nucleotides, but we have RNA nucleotides right here. All right, so what we want to do is we want to make an mRNA molecule, but we need this process to get started first. That's going to be with initiation. All right, so what we have here is our TATA box. All right, just TATA, and that allows a complex of transcription factors to bind right here. All right, this is what we call our promoter region, right, to get this process started of transcribing our molecule. All right, again, here we have our transcription factors. All right, now also kind of part about starting this process is this could be sometimes like tens of thousands of nucleotides or bases down, like upstream, is we could have these enhancers. Right? And as we've seen in that like animation, is sometimes what'll happen is that enhancer will literally fold over, touch here, and then boom, the process goes into place. Right? But here's our promoter region, and what's going to happen is this is going to essentially allow RNA polymerase to bind here. This is essentially the start of our transcription and factor initiation complex. All right. Once everything's set into place, we are now going to go into our process of elongation. All right. RNA polymerase is going to start just a little bit further down from our promoter region. All right. In this case, we're going to start right where our DNA opens up. All right. RNA polymerase is going to actually break those hydrogen bonds. And while it's breaking those hydrogen bonds, it's also adding in nucleotides. So here, if I have a T, I'm going to add in an A, right? If I have a G, I'm adding in a C, and so on and so forth. It's running down the line right here, all right? Now, in terms of the direction that it's going, it's adding nucleotides from the 5 to 3 direction. So here, my DNA is 3 to 5 going this way. So then it's going to be 5 to 3, all right, going this way for my RNA molecules, all right? Now, to speed this process up for both you guys and the video, I already have my mRNA molecule that would get made from this template strand right here, right? So RNA polymerase is adding in these base pairs, right? This is eventually going to leave right here, all right? Now, this is where we're getting towards so let's just say our RNA polymerase is still doing its thing right around here. I need to kind of move this down a little bit so we could see it. All right. This is where we're going to get to the termination phase. All right. What's going to happen here is this RNA polymerase, all right, when it gets closer to like this, essentially this A, where are we going with it? All right. Once it's making this AAUAAA sequence right here, all right, a little bit further down, we're going to end, all right, our transcription, all right, and what's going to happen is, all right, a little bit downstream from this AAUAA, this is what we call our polyadenylation signal, all right, it's going to cause some proteins, some enzymes down the line to cleave just a little bit down the line right here, all right, and we have now made our mRNA molecule, or rather should I say we've made our pre-mRNA molecule, because now we need to process it. So let's take the DNA out of the way, all right? So here we have pre-mRNA, all right? Oh, by the way, kind of going back here, forgot this. This area right here, right, where we had like that polyadenylation signal, right, this area where it's going to cause this termination phase. This is called the terminator region right here of our DNA, okay? So, taking the DNA out of the way, here we have our pre-mRNA, and what we're going to do now is some RNA processing, okay? So what's going to happen here is we are going to, I just want to make sure everything's in the scene. All right, looks like we could just about get it. All right, maybe need to go back a little bit. All right, we're going to have a 5 cap. All right, come on right here. Now, technically, this came on like pretty quick as RNA polymerase was making its nucleotides. This gets added right away. So this is our 5 cap. 5 cap, it's essentially a guanylene with these extra phosphates on it. All right, now, what are the 3 functions of this 5 cap, all right, number one, it allows export out of the nucleus, right? So essentially this is going to bind to like a nuclear pore area, and that just essentially says, hey, you're cool to let this thing leave the nucleus. So that's one. Number two is it prevents degradation like outside where there could be exonucleases that are cutting things down. And number three, it allows, this is going to bind to that ribosome to get this process of translation started once it's out in the cytoplasm. So 3 functions of that 5 cap, all right? Now here on our RNA, we also have on the other end, we have a poly A tail, right? It's really just going to be at the end right there, and I know you can't, it's right over there, right? There's our poly A tail, all right? Now, the function of our poly A tail, I don't know where the card is, but the function of our poly A tail, all right, is essentially to prevent degradation by exonucleases. So you can imagine there's some exonucleases maybe out in the cytoplasm, all right, that are eating up the RNA, all right? If it's just getting rid of these A's, it's totally fine, right? We don't want the exonucleases to take care of the stuff that's going to be coding, so it's adding a layer of protection there, all right? Some other things with our RNA processing, all right? Let's just identify some of our regions, all right? Now, you'll have noticed on the DNA there was this like shaded in area. TAC, what's going to bind there is AUG. AUG, where are we right here? All right, that is my start codon. I just can't find the card, all right? But that's going to be my start codon, AUG, all right? Where that's going to code for methionine, and we're going to get some amino acids right there. So that means that everything to the left in between that AUG here is going to be the 3 untranslated region of my mRNA, right? This is not going to get translated, right? Which means this is my protein coding segment, AUG, all the way down the line until if we get to that DNA right here, right, that ATC would give me UAG, so that UAG right there would be my stop codon, right? So that's the protein coding segment. Once we get past that UAG, we are now at the 5' UTR region, and I just made a mistake. This is the 5' UTR region, right? Makes sense because this is the 5 end of my RNA, 5 cap, right? Mistake. Over here is our 3' untranslated region, and then we get to the poly A tail, all right? So that's part of RNA processing. Really what we've done is we've created a cap, but then we're going to get into some really interesting thing where we're going to start to make exons and introns, all right? So interestingly enough, all right, technically you may not even call this the protein coding segment yet because there's going to be parts of here that aren't going to code for a protein, right? Here's my sequence right here for mRNA, right? And what's going to happen is we're going to have some spliceosomes come by, and they are going to cut, all right, specific regions, all right? The way that I have it right here is here we have our start codon, all right, and AUG, GUC, AG. So that means right here we need to cut. And again, what's going to be doing that is this spliceosome, all right? Now over here, we're going to start again with UAC, all right? So that's going to be, we want that in our mRNA sequence. So I'm going to cut right here, all right? And after this G, we're going to cut. All right, again, that's the end of that sequence I want, and then the beginning of the sequence I want is this AUU. So I'm going to cut right there. All right, so what are these things? All right, these things we want to cut out right. Where am I missing? UAC. Okay, right here we want to cut these things out. These things are called introns because they are going to stay in the nucleus. They'll eventually get degraded by some exonucleases, all right? And then we also have our exons. These are exons because they are going to exit the nucleus, go to the ribosome for translation, all right? So this is going to form here, 5 cap, poly A tail, all that stuff, all right? Technically now I would call this the protein coding segment of our RNA molecule, all right? Now, what this allows for, too, is what's called alternative RNA splicing. I could have the exact same gene, But maybe the spliceosomes only cut out that second section and keep this section, which means I'm going to get this is now part of the new protein coding segment. I'm going to get a new protein. Or maybe the spliceosomes don't cut it at all, and so I get another protein from the same gene. Or maybe it cuts out this part and keeps this part in. Essentially what I'm able to get is one gene, but a bunch of different proteins, polypeptides that I could get from that one gene based on how the spliceosomes are processing this RNA, all right? So let me take away some of the stuff over here, all right? So essentially we have it's no longer pre-mRNA, it is just straight up mRNA, all right? This mRNA is going to leave out of the nucleus, it's gonna go to the cytoplasm, all right? And it's gonna go to a ribosome. I know I got a card, don't know where it is right now. It's gonna go to a ribosome for the process of translation. Again, that 5 cap is gonna bind to that ribosome, start that process. Thanks for watching.